Deinococcus radiodurans, a bacterium that might live on Mars and is more metal than you. Deinococcus radiodurans, a bacterium that might live on Mars and is more metal than you. Published on the 8th of November 2022, written by Francis Davies under the categories of microbiology and news. This reading focus comes to us from biotechniques.com. Biotechniques, the International Journal of Life Science Methods. There could be life on Mars. It could be pretty hardcore. It could be coming to Earth. There is life on Earth. It could be going to Mars. Scientist frown. Welcome. You're listening to Biotechniques Reading, presented by Hakeem Alibokas Alexander for World Reading Club in association with Uniquilibrium. Biotechniques continues. You're never going to be more metal than a tardigrade. Anything that can casually drift through the vacuum of space like it's Sunday at the beach is automatically worth serious respect. But you've almost certainly got something like bacteria beat, right? While we've long since abandoned the search for little green men or portals to hell on Mars, researchers at Northwestern University, Illinois, United States of America, are suggesting that life is possible on the red planet. What's more, they're tentatively flagging the possibility of microbial cross-contamination between our planet and Mars. Prospective plans and missions to the fourth planet are under increasing focus from industrial interests, running at cross purposes to the scientific community, which holds the following two governing principles of protection. The first is that spacecraft should not carry terrestrial biological materials that could compromise future studies. This aims to prevent forward contamination. The second is that Earth should be protected against the introduction of microorganisms returning from extraterrestrial bodies. This aims to prevent backwards contamination. This concern may seem slightly excessive, given the fact that it is directed at a planet with a surface comprised largely of basalt rock covered in ferric oxide dust and an outer mantle of which roughly 18% is iron, according to one 2004 estimate. Or, considering the temperature, which averages a slightly nippy negative 63 degrees Celsius in the mid-latitudes, if that isn't too much for you, try the unremitting bombardment of galactic cosmic radiation and solar protons, which is to say that surviving on Mars is perhaps a bit beyond the survival capabilities of the average person, or, indeed, the average anything. But as an esteemed philosopher once observed, life, finds a way. Previous studies had confirmed the veracity of this assertion, identifying a microbe nicknamed Conan the bacterium, or actually Conan the bacterium. Previous studies had confirmed the veracity of this assertion, identifying a microbe nicknamed Conan the bacterium that could potentially survive on Mars for 1 million years. In order to further explore the potential of life on Mars, scientists at Northwestern University have run a set of simulations to put the mettle of Conan and other microbes to the test. If there are bacteria present on Mars, they would have to be incredibly sturdy, capable of surviving close to the surface despite the inhospitable conditions. To assess the capability of existing microbes on Earth, to withstand these conditions, the researchers worked out the survival limits of microbial life to ionizing radiation. After this, they exposed 6 forms of earth bacteria and fungi to the simulated conditions of a frozen and dry Martian environment, complete with gamma rays and protons to replicate radiation in space. Biotechniques suggest that you read also Microorganisms in a Volcanic Crater Lake. Microorganisms in a volcanic crater lake sheds light on life on Mars. The biochemical capabilities of microorganisms from the Laguna Caliente, a hydrothermal crater lake, was studied using DNA sequencing and gives insight into how life on Mars could survive. You've been listening to Biotechniques Reading, presented by Hakeem Alibokis Alexander for World Reading Club in association with Uniquilibrium. Biotechniques Reading continues. The outcome of these tests confirmed that it is in fact possible for a select few terrestrial microorganisms to survive a Martian climate over the course of hundreds of millions of years. Deinococcus radiodurans, the formal moniker of the previously noted Conan, is one such bacterium. Shrugging off radiation and temperature and outlasting bacillus spores, which are known to survive for millions of years here on Earth. It was impressive enough for Deinococcus radiodurans to last several hours on the surface while exposed to ultraviolet light. When buried just 10 centimeters below the surface, the survival period lasted an adequate 1.5 million years. When buried 10 meters below the surface, Deinococcus radiodurans could survive for a cool 280 million years. To put that in terms that a human might have some vague hope of grasping, 280 million years is just shy of the span of the entire Paleozoic era, which was divided into 6 separate geologic periods. At the point where you're measuring time in rock formations, it's time to give up. This titanic toughness is partially due to the genomic structure of the bacterium, with chromosomes and plasmids perfectly aligned and linked, making them repair-ready after a tanning session that would make Patrick Bateman blush. All of which means that if a microbe similar to this bacterium evolved during a period in which water flowed on the planet, then there is a possibility that its living remains could be resting, Cthulhu-like, deep below the surface of Mars. The findings reinforce the idea that remnants of Martian life, if such a thing ever existed, could be uncovered by investigations by 2 future missions, ExoMars and the Mars Life Explorer, which we will drill into the Martian soil and gather minerals from 2 meters below the surface, highlighting the risk of reverse contamination. These simulations also suggest that some strains of bacteria are capable of surviving in the harsh environment of Mars, opening up the possibility that the planet itself could accidentally become contaminated by bacteria traveling with future astronauts and space tourists. Source: Horn W.H., Volp R.P., Corza G. et al. And others. Effects of desiccation and freezing on microbial ionizing radiation survivability. Considerations for Mars sample return. Astrobiology 22, page 11, 1337. Well, actually, Astrobiology 22. I don't know what this is. Chapter 11? Anyway, it looks like it's page 1337 to 1350. It's from 2022, and there's a press release. You've been listening to Biotechniques Reading, presented by Hakeem Alibokis Alexander for World Reading Club in association with Unequilibrium. This press release comes from Northwestern University, northwestern.edu. Ancient bacteria might lurk beneath Mars' surface is the title. New study finds the chances of uncovering life on Mars are better than previously expected. Published on October 25th, 2022 by Amanda Morris. Researchers believe the Conan the bacterium pictured could survive for 280 million years on Mars if buried. This article is published under the categories of life sciences, space, and Weinberg College. When Mars' first samples return to Earth, scientists should be on the lookout for ancient sleeping bacteria, a new study has found. In a first-of-its-kind study, a research team, including Northwestern University's Brian Hoffman and Ajay Sharma, found that ancient bacteria could survive close to the surface on Mars much longer than previously assumed. And when the bacteria are buried and thus shielded from galactic cosmic radiation and solar protons, they can survive much longer. These findings strengthen the possibility that if life ever evolved on Mars, its biological remains might be revealed in future missions, including ExoMars, Rosalind Franklin rover, and the Mars Life Explorer, which will carry drills to extract materials from 2 meters below the surface. And because the scientist proved that certain strains of bacteria can survive despite Mars' harsh environment, future astronauts and space tourists could inadvertently contaminate Mars with their own hitchhiking bacteria. The paper was published today, October 25th, in the journal Astrobiology. So reading in context, it said then today. It is now November 8th, 2022, at the time of this reading. Continuing, northwestern.edu continues. Our model organisms serve as proxies for both forward contamination of Mars as well as a backward contamination of Earth, both of which should be avoided, said Michael Daly, a professor of pathology at Uniformed Services University of the Health Sciences, USU, and a member of the National Academy's Committee on Planetary Protection, who led the study. Importantly, these findings have biodefense implications too, because the threat of biological agents such as anthrax remains a concern to military and homeland defense. We concluded that terrestrial contamination on Mars would essentially be permanent over time frames of thousands of years, said Hoffman, a senior co-author of the study. This could complicate scientific efforts to look for Martian life. Likewise, if microbes evolved on Mars, they could be capable of surviving until present day. That means returning Mars samples could contaminate Earth. Hoffman is the Charles E. and Emma H. Morrison Professor of Chemistry and Professor of Molecular Biosciences in Northwestern's Weinberg College of Arts and Sciences. He is also a member of the Chemistry of Life Processes Institute. Simulating Mars. The environment on Mars is harsh and unforgiving. Arid and freezing conditions, which average -80 degrees Fahrenheit, -60 degrees Celsius, at mid-latitudes, make the red planet seem inhospitable to life. Even worse, Mars also is constantly bombarded by intense galactic cosmic radiation and solar protons. To explore whether or not life could survive in these conditions, Daly, Hoffman, and their collaborators first determined the ionizing radiation survival limits of microbial life. Then they exposed 6 types of earthling bacteria and fungi to a simulated Martian surface, which is frozen and dry, and zapped them with gamma rays or protons to mimic radiation in space. If microbes evolved on Mars, they could be capable of surviving until present day. That means returning Mars samples could contaminate Earth. Brian Hoffman, chemist. There is no flowing water or significant water in the Martian atmosphere, so cells and spores would dry out, Hoffman said. It also is known that the surface temperature on Mars is roughly similar to dry ice, so it is indeed deeply frozen. Ultimately, the researchers determined that some terrestrial microorganisms potentially could survive on Mars over geologic timescales of hundreds of millions of years. In fact, the researchers discovered that one robust microbe, Deinococcus radiodurans, affectionately known as Conan the bacterium, is particularly well suited to surviving Mars' harsh conditions. In the novel experiments, Conan the bacterium survived astronomical amounts of radiation in the freezing, arid environment, far outlasting bacillus spores, which can survive on Earth for millions of years. Radical radiation. To test the effects of radiation, the team exposed samples to large doses of gamma radiation and protons, typical to what Mars receives in the near subsurface, and far smaller doses, which would occur if a microorganism was deeply buried. Then, Hoffman's team at Northwestern used an advanced spectroscopy technique to measure the accumulation of manganese antioxidants in the radiated microorganism cells. According to Hoffman, the size of the radiation dose that a microorganism or its spores can survive correlates with the amount of manganese antioxidants it contains. Therefore, more manganese antioxidants means more resistance to radiation and more enhanced survival. 280 million years that Conan the bacterium could survive buried on Mars. In earlier studies, previous researchers found that Conan the bacterium, when suspended in liquid, can survive 25,000 units of radiation or grays, the equivalent to about 1.2 million years just below Mars' surface. But the new study found that when the hardy bacterium is dried, frozen, and deeply buried, which would be typical for a Martian environment, it could weather 140,000 grays of radiation. This dose is 28,000 times greater than what would kill a human. Although Conan the bacterium could only survive for a few hours at the surface while bathed in ultraviolet light, its lifetime improves dramatically when it's shaded or located directly below Mars' surface. Buried just 10 centimeters below the Martian surface, Conan the bacterium's survival period increases to 1.5 million years. And when buried 10 meters down, the pumpkin-colored bacterium could survive a whopping 280 million years. Looking to future missions. This astonishing survival feat is partially thanks to the bacterium's genomic structure, the researchers found. Long suspected, the researchers discovered that Conan the bacterium's chromosomes and plasmids are linked together, keeping them in perfect alignment and ready for repair after intense radiation. That means that if a microbe similar to Conan the bacterium evolved during a time when water last flowed on Mars, then its living remains could still be dormant in the deep subsurface. Although radiodurans, that's Deinococcus radiodurans, or Deinococcus radiodurans, although Deinococcus radiodurans buried in the Martian subsurface could not survive dormant for the estimated 2 to 2.5 billion years since flowing water disappeared on Mars, such Martian environments are regularly altered and melted by meteorite impacts, Daly said. We suggest that periodic melting could allow intermittent repopulation and dispersal. Also, if Martian life ever existed, even if viable life forms are not now present on Mars, their macromolecules and viruses would survive much, much longer. That strengthens the probability that, if life ever evolved on Mars, this will be revealed in future missions. The study, Effects of Desiccation and Freezing on Microbial Ionizing Radiation Survivability, Consideration for Mars Sample Return, was supported by the Defense Threat Reduction Agency, grant number HDT RA1620354, and the National Institutes of Health, grant number GM111097. For journalists, view the news release for media context and assets. You've been listening to Biotechniques and Now Northwestern.edu reading, presented by Hakeem Ali Bokus Alexander for World Reading Club in association with Uniquilibrium.